Now let's talk about replication, finally. So what do we know now? We know that replication occurs at interphase, it occurs at the S phase. So now let's take a look at how replication actually occurs. And I'm just going to simplify this replication process by drawing the DNA strands in a linear fashion. Please keep in mind, these two strands are technically intertwining with each other. I have hydrogen bonds, remember, they have a helical structure. But if I draw it like this and try to explain it to you, it's going to take forever. So what am I going to do? I'm going to explain it to you in a linear fashion. So it's going to be simple. So here, this is, once again, this DNA strand is running in an anti-parallel direction, 5' to 3', 5' to 3'. Please remember this. This is very, very important. Now, I want to actually start labeling this. I'm going to add some random nitrogen spaces, and this serves as basic practice for those of you who are taking the regions. And for those of you who are taking the AP exam, I mean, this should be a breeze for you. You must know how to do this already. So, I'm just going to label anything A, C, C, G, T, A, C, G, T, A, C, C. The complementary strand will contain the bases that correspond to this. Remember the DNA base pairing rule? A pairs with T and C pairs with G. So therefore A pairs with T, C pairs with G, C pairs with G, and so on and so on and so on and so on. So this is fairly easy. Great. So I have my DNA strand, but once again, this is oversimplified. I'm only showing you this much. I'm going to teach you about replication, but once again, replication we have thousands and thousands and thousands of nucleotides, so this is obviously oversimplified. So I'm just going to show you how replication works first in this way, and then I'll get a little bit more technical. So, let's see. The first thing we need in order to replicate our DNA strand is an enzyme called Helicase. So, number one, we need Helicase, and Helicase is an enzyme that unwinds and breaks apart the hydrogen bonds between the strands. So this double-stranded molecule is held together by hydrogen bonds. A and T have 2 hydrogen bonds, C and G have 3 hydrogen bonds, and we have to break them apart. Helicase is going to do that. And you might say, hey, why do we want to break it apart? Why do we care? Well, we need to break it apart so we can allow a bigger enzyme called DNA polymerase 3 to ultimately enter into each of these strands and start synthesizing the new complementary strand. But we'll talk more about that in a bit. So Helicase is going to start to unwind the DNA. And I'm going to just pretend that, okay, you know what? Helicase is going to start over here, let's pretend. It's going to start unwinding in this side right here. This would be noted as my origin of replication. It's my replication fork. Now, the important thing to understand about DNA replication is that DNA replication is bi-directional. What does that mean? DNA replication is bi-directional, meaning you can start going this way, or you can also go this way. It actually goes both ways. But if I start to show you in a bi-directional manner, it might get a bit too confusing. So I'm going to show you first the simple way, and then I'm going to teach you the bi-directional way, so it's much easier for you to understand. So just remember, DNA replication is bi-directional, but now let's see what happens. Let's just pretend for now that, in fact, this DNA strand, okay, so Helicase, let's pretend, is going to start from here. Helicase, I'm going to label Helicase with a green marker. Here's my Helicase enzyme. We're going to pretend that replication is going this way, so Helicase ultimately is going to start unwinding this way. And so it does. Helicase starts to unwind this. Break it apart, break, break. And ultimately, what do you see? Ultimately, you will see something like this. Five prime, 3 prime, 5 prime, 3 prime. It is very crucial for you to always label your strands, or it can get very confusing very fast. Always label everything. Now, why did I just stop right here? Well, I want to show you what happens. Now, Helicase works at a very fast pace. DNA replication, all the things actually work together. So in order to explain replication, I'm going to just freeze Helicase in time, and I'm going to explain what happens here right now. So let's just pretend Helicase only broke apart this much so far. Helicase still has to break apart this hydrogen bond right here, but let's just say it hasn't yet. We freezed it. Now let's take a look at what actually happens to this side. Well, let's just pretend we have a... Okay, so here's my C, A, T, and G. Therefore, my complementary strand is going to have a C, A, T, G, G. Now, something very cool actually happens. Helicase also works with a very special enzyme called topoisomerase. And I think topoisomerase is very, very fundamental. Topoisomerase and Helicase essentially work together. Topoisomerase. And what this enzyme does, it creates either a single-stranded or a double-stranded break to reduce the tension. Now, what does that mean? Well, let's take a look at this. You ever played with a spring? You know, those springs that are found in your pens, right? These little spring things. I like to look at a spring. Imagine I started to untwist one side of this spring. What would happen to the other side? Am I increasing the pressure? Am I increasing the tension? Yes, there's much more tension in that area. As I'm breaking apart the hydrogen bonds, as I'm untwisting this supercoiled DNA, what's happening is that there's a lot of tension building up in this region. Do I want a lot of tension amongst? I have I have my DNA bases which code for genetic instruction. Do I want heavy tension with this? I might do something like mutate them, right? So what happens? Topoisomerase comes in and it tries to reduce the pressure. How does it do that? How does it reduce tension? Well, it's going to cut the DNA, and by cutting the DNA right here, so let's just say we've unwinded this, topoisomerase will come in and cleave the DNA right here really, really fast, and then religate them back together. Why would you want to reconnect them back together? Well, you still have to go on, right? It just doesn't cut it, and that's it. You cut it, and then you religate it. So you reduce the pressure. So you cut the DNA, and then religate. This will reduce the tension. So Helicase and topoisomerase work together. As Helicase is unwinding and breaking apart the hydrogen bonds, topoisomerase also comes in and starts to reduce the tension that is being built up because of Helicase. So that's good. So far we have Helicase and topoisomerase. Let's see what happens next. The next enzyme you should know about, or the next protein you should know about, is something called SSBs, single-stranding binding proteins. What is that? Single-stranding binding proteins. Single-stranded binding proteins. Sometimes on your exam you will see it noted as SSBs, and these guys will attach to one strand to prevent reannealing. What does this mean? Attach to one strand in our double-stranded DNA sequence to prevent reannealing of the strands. Well, as I just told you, Helicase is going to break it apart, but notice, G and C, T and A, they still have some kind of attraction. They can accidentally reconnect, right? They can accidentally reanneal. Do you want that? Do you want to reanneal? Why do you have Helicase in the first place? Helicase makes sure that the hydrogen bond is broken. You want to assure yourself that these guys won't reconnect back together. So this single-stranding binding protein will wrap around one of the strands. It's going to wrap around one of the strands to prevent reannealing. So you have some weird-looking thing wrapping around, and no longer can you reanneal. So that's good. The next thing you want to learn, the next thing we should learn about, the most important enzyme, which is DNA polymerase 3. The DNA polymerase 3 is your key player here. This guy is going to come in and synthesize the complementary strand. This guy's going to come in and say, okay, I'm going to make a new strand here. Make the complementary base. What's the complementary base of C? G, G, T, A, C. It's going to do that. Same thing here. But it's not that simple. DNA polymerase III is somewhat of an odd enzyme. It doesn't know where to go. It's not as smart. DNA polymerase III only recognizes primers. DNA polymerase III recognizes primers. And what are primers? Well, primers are a short segment of RNA. So what are primers? Primers are a short segment of RNA. And once we make a primer, DNA polymerase III can finally say, oh hey, I recognize you, primer. Let me go ahead and bind to you and now synthesize the complementary strand. Now there's something very cool about this DNA polymerase III. This guy only synthesizes from the 5' to the 3' region. It only travels from 5' to 3', 5' to 3'. Get used to this notion. This is very, very important when we talk about transcription and RNA polymerase. These enzymes always synthesize from the 5' direction to the 3' direction. This is probably going to be one of your exam questions. DNA pol 3, which way does it synthesize a complementary strand? 5' to 3'. So okay, DNA polymerase III needs to recognize a primer. I just told you primers are a short segment of RNA. And now we can talk about how primers are made. There's actually an enzyme called a primase. So primase is an enzyme that will make a primer. So primase makes a primer. DNA polymerase III can recognize the primer and then synthesize from the 5' to 3' fashion. But now, okay, you might say, hey, where are we going to put this primer? And it's very, very simple. I'm going to show you exactly what to do now. I'm going to remove this single-stranded binding proteins to make this diagram less complex. Let's see what happens. I need to put a primer in this strand. I need to put a primer on this strand. Remember, I have to replicate both these strands. Here's the thing. Wherever you see a 3', you're going to put the primer over here. The primers will always be labeled as the 5' region. The primers will always be labeled as the 5' region. I'm going to actually make it a different color. I'm going to make it green so it's clear. Here's my primer. This is going to be my starting of the new strand. And I'm going to do what? This is going to allow my DNA polymerase III to come in. And notice, this is my 5' region. That means this is my, what? 3' end. This is my 3' end. 5' to 3', 5' to 3', 5' to 3', 5' to 3'. Anti-parallel, once again. Look, 5 to 3, 5 to 3. Does that make sense? Is that starting to make sense now? So DNA polymerase III comes in and says, hey, okay. And it starts to synthesize the complementary strand, complementary base G, G, T, A, C. Great. How about over here? Well, I told you that primers will be labeled as 5'. Can I put a primer over here? Is that possible? Can I put a primer over here and label this 5'? Does this match what we've learned so far? 5' and 5'. No. 5'3". This has to be what? 3'. So can a primer go here? No. So this will automatically by default be 3', but where will the primer be? Well, some of you might say the primer should be here, right? But because this is a 3', so if we put a primer here, it's 5'. But these two strands still have hydrogen bonds between it. We're not going to focus on these. We're going to focus on the part that has been broken apart by Helicase. So over here, technically now, this side over here can be labeled as the 5' end. So I'm going to make a primer over here, and I'm going to label it 5 prime. And guess what? DNA polymerase III once again will come in and do what? Synthesize a complementary strand from the 5 prime direction to the 3 prime direction. Very simple. What's it going to make? C, G, A, T, T, A, G, C, G, and C. So, simple enough, right? Once again, you should know how it works with the primers. Very simple. Always see if the pattern fits. 5 to 3, 5 to 3, 5 to 3, 5 to 3. Always in the anti-parallel direction. That's all you have to know now. But now what happens? Now what happens? Well, Helicase is going to still unwind. Now you might say, hey, why are you teaching it to me in such a way that Helicase unwinds right here, and why are we stopping here? Why don't we just learn when Helicase stops over here? Well, you have to realize that our DNA bases there's many bases. If we did this, if Helicase just said, okay, so if this is our entire genome, and we have to replicate this entire thing, and Helicase said, okay, everybody wait, DNA polymerase III wait, primase wait. My name is Helicase, and I'm going to unwind the entire genome. It's going to take forever, and then topoisomerase will also follow, and then we have primase that will lay down a primer, and so on and so on. Remember, we have to work very, very fast, and replication is really an efficient process. It's a remarkable process because, you know what we have? Helicase comes in, topoisomerase follows, and almost immediately, as we're doing that, single-stranded binding proteins come in, primers are already laid down, DNA polymerase III comes in, and as Helicase keeps unwinding the DNA, once again, single-stranded binding proteins wrap around the new broken apart hydrogen bonds between the both strands. We have new primers laid down, and so on and so on. So it's much more efficient. So let's see what happens now. So let's see how that actually works. Okay, so now let's pretend that Helicase has broken apart this as well. So up to here. So let's draw that. 5 prime region, 3 prime, 5 prime, 3 prime. I'm not going to actually label now because I don't want to waste too much time, but I want you to get the main idea. So far, what have you done so far? Here's my primer. We're going to label it the 5 prime region. And what have I done? I've synthesized about this much. Let's pretend. I know it's not proportional. And how about this one right here? We have a primer right here. And we are going in this direction, right? We've synthesized so far here. Let's label this 5 prime, 3 prime, 5 prime, 3 prime, 5 prime, 3 prime. Always match the pattern. And your life will be easy when it comes to replication. Great, now let's see. Whoa. We have now broken apart more between this double-stranded DNA. How are we going to replicate? Once again, DNA polymerase III synthesized in the 5 prime to the 3 prime fashion. It only reads and synthesizes from 5 prime to 3 prime. Well, here's my DNA polymerase also, right? So it's over here. It synthesized a complementary strand. It's waiting over here patiently. And guess what it's going to do? Remember I told you Helicase is going this way. I told you that DNA replication is bi-directional, but we're only focusing right now on this direction. So guess what? It's still going from the 5 prime fashion to the 3 prime fashion. So DNA polymerase III is just going to keep continuing and make it. We have to do nothing fancy here. It's always going this way. So as long as I'm breaking apart my hydrogen bonds with Helicase, DNA polymerase III can just easily synthesize the complementary strand, make the new one, make the new base, complementary base, so on and so on and so on and so on. Easy. How about this though? This is my 5 prime region, right? Primer. How is DNA polymerase III traveling here? This way. This direction. It's going this way. Can I now magically go this way? I just told you DNA pol 3 only synthesizes from the 5' to 3' fashion. How are we going to do this? Well, any ideas? Hopefully you've come to the conclusion that we need more primers. We need to set more primers. If I set another primer over here, guess what? If I put a primer over here, now this will become my 5' region. And what will happen? DNA pol 3 will come in and it's going to synthesize from the 5' direction to the 3' direction. Do you see that? We have multiple primers on the bottom strand. We have multiple primers on the bottom strand. And then, when Helicase breaks this apart, what do you think is going to happen? This guy is simply going to keep going this way, and we're going to have to add another primer over here. And another DNA pol 3 will come in and synthesize in this fashion. Not so hard. Well, we have special names for this. You see this top strand where it's just nicely flowing. We only have one primer and DNA pol 3 just goes on. This right here is called the leading strand. This is called the leading strand. And this right here, this strand where we have multiple primers, this is called the lagging strand. It's called the lagging strand, and each of these fragments are referred to as the Okazaki fragments. Each of these fragments are referred to as the Okazaki fragments. So you should know that. Lagging strand, leading strand. These are Okazaki fragments. Only the lagging strand contains Okazaki fragments. The leading strand does not. Please know this. In the next video, we're going to continue on talking about replication.